Hi. Cellular adaptation is the ability of cells to respond to various types of stimuli and adverse environmental changes. These adaptations include hypertrophy, which is the enlargement of individual cells, hyperplasia, which is the increase in cell number, atrophy, which is the reduction in the size and the cell number, metaplasia, which is transformation from one type of epithelium to another, and dysplasia, which is disordered growth of cells. An organ is in homeostasis with a physiologic stress placed on it. For example, the heart functions by pumping against the systemic blood pressure. However, if there is an increase, a decrease, or a change in stress on the heart, it could result in growth adaptations. Now, growth adaptation could manifest in different ways depending on the cellular properties of the tissues involved. Let's look at hyperplasia and hypertrophy. So in hypertrophy, there is an increase in cell size, whereas in hyperplasia, there is an increase in cell number. Hypertrophy involves gene activation for protein synthesis and the production of organelles. On the other hand, hyperplasia involves the production of new cells from stem cells. Hyperplasia and hypertrophy generally occur together. A good example is the enlargement of the uterus during pregnancy. Now, a deviation from this rule is a case of permanent tissues, example, the cardiac muscle, skeletal muscle, and nerve. These tissues cannot make new cells and therefore undergo only hypertrophy. For example, in a patient with systemic hypertension, the cardiac myocytes undergo only hypertrophy. Now, hyperplasia could either be physiologic or pathologic. For example, in women, the endometrium undergo physiologic hyperplasia every month during the menstrual cycle due to estrogen activity. However, sometimes the uterus undergoes pathological hyperplasia. This can progress to dysplasia and eventually into cancer. Now, the endometrium grows with exposure to estrogen and sheds with the exposure to progesterone. However, if the endometrium is exposed to excessive estrogen for a prolonged period of time, it can undergo endometrial hyperplasia and eventually to endometrial carcinoma. A notable exception to this rule as well is the case of benign prostatic hyperplasia, which does not increase the risk for prostate cancer, hence the name benign prostatic hyperplasia. Now let's move on and talk about atrophy. So atrophy occurs when there is a decrease in stress of an organ. For example, when there is a decreased hormonal stimulation, disuse, or decreased nutrients and blood supply, there will be a decrease in organ size. This occurs through decrease in the size and the number of cells. Decrease in cell number occurs via apoptosis. Decrease in cell size, on the other hand, occurs via two different pathways. The first of these is the ubiquitin proteasome degradation of the cellular cytoskeleton. The second pathway is autophagy of cellular components. Now let's illustrate this briefly. So in ubiquitin proteasome degradation, intermediate filaments of the cytoskeleton are tagged with ubiquitin and destroyed by proteasomes. Autophagy of cellular components involves the generation of autophagic vacuoles. Now these vacuoles fuse with lysosomes whose hydrolytic enzymes break down the cellular components. So now we'll move on to talk about metaplasia. So in metaplasia, a change in the stress on an organ leads to the change in cell type. Most commonly, it involves a change of one type of surface epithelium into another. For example, squamous epithelium, columnar epithelium, or urothelium. Now, urothelium is found in the urogenital tract. Metaplastic cells are better able to handle the new stress. An example is in the case of Barrett esophagus. Now, the esophagus is normally lined by non-keratinizing squamous epithelium. This lining is suited to handle friction of food bolus. Now, acid reflux from the stomach causes transformation or metaplasia of the non-keratinizing squamous epithelium to a non-ciliated mucin-producing columnar cells, which is better able to handle the stress of the acid. Now, metaplasia occurs through the reprogramming of stem cells at the site of the stress, which then produce the new cell type. But the good thing is, metaplasia is usually reversible when the driving stressor is removed. So for example, treatment of a gastroesophageal reflux may reverse the Barrett esophagus. Now, under persistent stress, however, metaplasia can progress to dysplasia and eventually result in cancer. For example, Barrett esophagus may progress to adenocarcinoma of the esophagus. There is an exception to this case, which is generation of apocrine metaplasia or the development of apocrine metaplasia. Now, apocrine metaplasia of the breast is seen in relation to fibrocystic changes in the breast, but it does not necessarily increase the risk for breast cancer. Now, another case of metaplasia has to do with vitamin A deficiency. Vitamin A is necessary for differentiation of specialized epithelial surfaces, such as the conjunctiva covering the eye. So, in vitamin A deficiency, the thin squamous lining of the conjunctiva undergoes metaplasia into stratified keratinizing squamous epithelium. Now, this change is called keratomalacia. Now, mesenchymal or connective tissues can also undergo metaplasia. A classic example is the case of myositis ossificans, in which the connective tissue within muscle changes to bone, so there is ossification of the connective tissue within the muscle. So this happens usually during the healing process after a trauma. Now let's talk about dysplasia. So dysplasia means disordered cellular growth. Most often it refers to proliferation of precancerous cells. For example, cervical intraepithelial neoplasia, usually abbreviated as CIN, represents dysplasia and it is a precursor to cervical cancer. Dysplasia often arises from long-standing pathological hyperplasia, for example, in the case of endometrial hyperplasia. Now, dysplasia could be reversed if the source of stress is removed. If the stress persists for a longer period of time, dysplasia could progress to carcinoma, in which case it becomes irreversible. And finally, let's talk briefly about aplasia and hypoplasia. Aplasia is the failure of cell production during embryogenesis. So an example of this is unilateral renal agenesis. Now, hypoplasia is a decrease in cell production during embryogenesis. This could result in small organ. So an example is a streak ovary formation in Turner syndrome. Thanks for watching. Please remember to like this video and subscribe to the channel. More videos coming up. See you in the next one.